Hello, my name is Amir. I'm a PhD student doing research in computational biology and bioinformatics. In this project, my focus is on using machine learning to design an efficient guide RNA for a gene editing tool called CRISPR-Cas9, which can be used for disease treatment. The main components of my talk will be about gene editing, CRISPR-Cas9, which is one of the methods for gene editing that I will use in my project, and then I'll talk about how this is related to cancer treatment and how machine learning could be applied to this process. So, why am I interested in gene editing? Well, gene editing is one of the methods that attracted scientists' attention, especially in recent years, as potentially one of the most effective solutions for curing diseases. But there is still concerns about potential consequences that are associated with gene editing due to the insufficient accuracy of the process being used that prevents the clinical level trials. Therefore, solving this issue will revolutionize the therapeutics at the clinical level and will hopefully have a worldwide health impact. So, what is gene editing? You may have heard of DNA or gene. DNAs are building blocks of our bodies, and these DNAs contain genes that have specific functions, like making our hair to have a specific color. Sometimes a mutation occurs in these genes, which can change their function and have harmful or useful results. Now, gene editing is the method that gives us the opportunity to make arbitrary changes to an organism's genetic code. There are four main techniques used for gene editing: restriction enzymes, zinc finger nucleases, Talon's gene editing, and then in 2012, scientists discovered a new method called CRISPR-Cas9, which is based on the defensive mechanism that existed for a long time in bacteria to fight off invading viruses. So why we chose CRISPR-Cas9? Well, CRISPR-Cas9 has made gene editing significantly cheaper, faster, easier, and more accessible compared to the existing methods. Using previous gene editing methods is often limited because they rely on proteins to guide editing machinery. Therefore, targeting each new genome site requires engineering and cloning of a new protein. In our bodies, we have strands of leather, just like DNAs, that are called RNA, with a few differences in structure and function from DNA. Unlike previous techniques, DNA recognition by the CRISPR-Cas9 system is specified by a guide RNA sequence, which makes this tool simple and facilitates the universal use. The CRISPR-Cas system has two main components: a guide RNA and a CRISPR-associated protein, or Cas, which has several versions isolated from different bacteria, and the most commonly used one is the Cas9. So how does the system work? The guide RNA is a specific RNA sequence that identifies the target DNA region of interest and directs the Cas protein there for editing. The Cas9 is directed to the specific DNA location and cuts the double strands of the DNA. Then a programmed DNA could be inserted in that area. Now, how it is related to cancer treatment? Well, we aim to target and repair the cancer-causing gene. And how are we going to do that? Well, the TP53 gene is the most frequently mutated gene in human cancer, with mutations found in about half of all cancer cases. Alterations in the TP53 gene are commonly associated with increased risk of cancer and may promote the division of cancer cells. Therefore, designing efficient guide RNAs to detect malfunctioning genes such as TP53 gives scientists the opportunity to make use of CRISPR technology at the clinical level for therapeutic purposes, especially for cancer treatment. So my main research idea is to design an improved guide RNA for gene editing using computational tools. The input would be sequences of DNA with different lengths, and it will output a guide RNA sequence of length between 17 to 23. We will consider different output length, different positions for components of the sequence, and different concentration of each component to achieve the best result. So far, different guide RNA design rules and tools have been developed for guide RNA design. These methods are categorized into different types, two of which are alignment-based methods and learning-based methods. The previous methods are related to my work as follows. The main similarity between my work and the previous works is that I'm also going to use machine learning methods such as deep learning. And the main difference is that I'm going to combine these methods with genetic algorithms to make a better design tool. Here we propose a learning-based tool for designing efficient guide RNAs using deep learning and genetic algorithms that takes an input DNA sequence, a genomic region, or a gene and their different features. We will measure accuracy of prediction by different criteria, one of which is mismatches of sequences that correspond to the desired target gene sequence, and the output will be potential guide RNA sequences with minimized off-target effects and maximized on-target efficiency. We assume that our model, which is based on machine learning, will perform better compared to other methods, such as alignment-based methods. Based on my literature review, I found that learning-based methods generally outperform alignment-based methods in terms of on-target efficiency. I also compared their performance in terms of specificity, and results indicate that learning-based methods, such as machine learning methods, usually introduce less off-target identification, which is desired for our research purpose, and therefore learning-based methods have better overall performance compared to other methods. During my research, I found that generally models don't perform well when trained on heterogeneous datasets, that is, datasets of different cell types. Also, I found that learning-based methods potentially outperform other methods. And lastly, we should care about the specificity more than efficiency, because targeting a wrong location in DNA can lead to very bad consequences, such as death of patients. Now, the remaining work is to improve an existing guide RNA design tool, especially in terms of decreasing off-target results, or to implement a new tool based on the findings from this research. The main point of my research was that we can make use of machine learning to improve the design of guide RNAs that are used in gene editing for disease treatment. Thank you.